dear chairs, organizers, mentors, students, dear young researchers, it is a great pleasure and honor for me to stand here today and present part of my journey in the realm of capsaicin and its receptor, and hopefully show you how it can be used as a potential tool in combating obesity. As you probably know, obesity develops if there is a disruption in the complex energy balance, which consists in part of energy intake in the form of food intake and energy expenditure in the form of heat production, which over the long term determine normal body mass. However, heat production is also in balance with heat loss, which regulate body temperature on the short term, and heat production can be achieved via two major ways: via shivering thermogenesis in skeletal muscle and non-shivering thermogenesis in brown adipose tissue. This latter tissue type was also shown in one of the slides of Csaba in the previous talks, and it is an important organ because it is dedicated to produce heat and consume the energy stores from the body, whereas the other adipose tissue type, white adipose tissue, is the one that stores the energy, and if it accumulates, then obesity develops. The role of brown adipose tissue was well established in small animals and newborns until 2007, when it was shown that also in adult humans there are substantial brown adipose tissue depots, which you can see as the black areas in this picture. And these depots become activated if adult humans are exposed to cold next to the spine, in the supraclavicular region, and in the interscapular region. So this shows the importance of the tissue in body temperature regulation, but it was also shown in other studies that over the long term, the amount of brown fat, which is now shown in red in these thermal images, contributes to the maintenance of normal body mass, because if it's active and present, then children remain lean. However, if its activity decreased in the supraclavicular region, that can be associated with obesity. And for the current talk, it is also important to note that mostly based on animal studies, it was shown that brown adipose tissue can be activated not only by cold, but also by some ligand substances, such as menthol, which then activates the sympathetic nervous system that results in an increased brown fat activity and energy expenditure. And of utmost importance, not only menthol, but capsaicin can also result in the activation of the same pathway and brown fat activation. So therefore, again, based mostly on animal experiments, it was shown that capsaicin can activate brown fat, while at the same time it can inhibit the accumulation of white adipose tissue, thereby prevent obesity in part by a decreased energy intake and also via some changes in microbiome. Capsaicin, as all of you probably know, is the pungent ingredient in hot chili peppers, and its concentration is often assessed on the Scoville scale, where the higher unit means higher pungency of the pepper, going from non-pungent to very highly pungent peppers like the habanero chili, which is in the Scoville heat unit range of approximately 100 to 300,000. And some newer peppers are already in the million range, while capsaicin itself is about 15 to 16 million Scoville heat units. It is important to mention that not only capsaicin, but other similar substances, the so-called capsaicinoids, can be also found in the hot peppers, which can have similar or somewhat decreased pungency compared to capsaicin. And if someone talks about paprika and peppers in Szeged, it must be also noted that not only capsaicinoids, but other substances like ascorbic acid can be also found in large amounts in pepper, as shown in 1937 by Albert Szent-Györgyi, who received the Nobel Prize 3 years later. But not only ascorbic acid, capsaicin was also heavily studied in Hungarian laboratories in Szeged and Pécs. The pioneering works of Professors Miklós Jancsó, Aranka Jancsó Gábor, János Szolcsányi, and Gábor Jancsó must be mentioned, the successors of whom are also prominent scientists. Some of them are sitting here with us today and also mentors of this program. And they summarized very nicely how this pioneering work of Hungarian scientists contributed to the Nobel Prize in Physiology or Medicine in 2021, which was a shared Nobel Prize awarded also to David Julius, who was portrayed on the journal Temperature in 2015, and received the award in part for cloning the receptor of capsaicin, which was formerly known as pain receptor, but then later it became the so-called transient receptor potential vanilloid 1, or in short, TRPV1 channel. The cloning was done in 1997, and it was also shown that not only capsaicin, but other painful stimuli, such as acidity or painful heat with a temperature above 42 degrees, can also activate the channel, which, if activated, results in a non-selective cation influx, and pain is often triggered, so its role in inflammatory and pain processes was clearly established. Later, thermoregulatory roles and roles in complex energy balance were also shown. And indeed, the cloning of the receptor made it possible to create mice genetically lacking the TRPV1 channel, so-called knockout mice. And if these mice were offered with habanero chili, they could eagerly consume it, even the hottest parts, the vessels and the seeds, without the sensation of pain. But as you probably know from your teachers and mentors, in all experiments there must be a control group, which was not so lucky in these experiments. They also tried to consume the pepper, but they soon gave up after a few attempts because of the development of pain. While we were studying the thermoregulatory phenotype of these mice, we found an unexpected surprising result in the vivarium, namely that when we looked at the mice colonies, we noticed that as a function of age, the body mass of the knockout mice, shown with the black symbols, significantly exceeded the body mass of the control mice, both in males and in female mice. And these animal findings took us to the continuation of the work, which was also the aim of the awarded paper, in which we wanted to see if the regular consumption of capsaicin diet could influence the blood fat levels also in humans. To answer our question, we used the tool meta-analysis, which is an ideal tool to summarize the available literature and available data in a certain field, and then quantitatively perform new data synthesis and provide support or rejection of a certain hypothesis. Therefore, it is also considered in some opinions as the highest, strongest scientific evidence in the hierarchy of scientific evidence. When we performed this meta-analysis, we raised the question whether consumption of capsaicinoids influence serum lipid profile in humans. So we collected studies in which blood cholesterol levels were compared between two groups of human subjects, one receiving capsaicinoid diet and the other on a control diet, and we checked different levels of cholesterol, the total cholesterol, which consists of both the bad cholesterol, LDL, or low-density lipoprotein, and the good cholesterol, HDL, or high-density lipoprotein, but we also analyzed the levels of these lipoproteins separately. So we created a search key that you can see on the left side here, and entered it in the three major databases, which identified nearly 5,000 papers, but after enabling filters, removing duplicates, and screening the titles and abstracts of these papers, altogether 10 full-text papers remained eligible for the inclusion of the data synthesis, which we did and used standard meta-analysis methods, which often result in the so-called forest plot that you can see in this slide. It looks complex, but in fact, it only shows each study in one row, and from each study, the extracted data of serum total cholesterol, in this case, from the control group and from the capsaicinoid treated group, from which the difference was calculated. It had to be standardized for variation and sample size, which gave us the so-called SMD, or standardized mean difference, that is also shown with the black square, and the arms of the square show the confidence interval, which is the variation of the parameter. If SMD is a negative value, then capsaicinoids reduced the parameter serum total cholesterol. If it's a positive, then they rather increased it. As you see in most studies, there was a decrease, and we also calculated the average SMD from two subgroups, namely groups which consumed capsaicinoids only, and another subgroup in which capsaicinoids were supplemented with other substances like tea extracts or caffeine. And finally, we also calculated the average from all studies, which average SMDs are shown as the diamonds. The vertical diagonal of the diamond shows the average SMD, and the horizontal diagonal is again the confidence interval. And the important part here is that in both subgroups and overall, there was a significant decrease in the serum total cholesterol level after the consumption of the capsaicinoid diet. But serum total cholesterol, as I mentioned, included both the good and the bad cholesterols, HDL and LDL. So these we also analyzed separately. And first, we checked LDL level. And what we found was, based on the same principles and methodology, as I mentioned before, that the average SMDs overall significantly decreased in case of the serum LDL cholesterol, which means that the drop in serum total cholesterol was caused, at least in part, by a decreased bad cholesterol level, LDL, due to the capsaicinoid diet. And we also wanted to see the good cholesterol, HDL, and here, in contrast with the previous results, we found that the SMD was near zero, so there was no significant effect on the good cholesterol after the consumption of capsaicinoids. And this takes us to the summary of our results, which says that the regular 1 to 3 month long consumption of capsaicinoids can reduce serum total cholesterol levels. And this decrease is mostly caused by the lowering of the bad cholesterol, LDL, whereas the good cholesterol, HDL, remains unchanged after the diet. And thus, our study provides novel quantitative support for the beneficial effect of capsaicinoid supplementation on the blood lipid profile in humans. And as for mechanism, it can be assumed that these beneficial effects are achieved through the activation of the sympathetic nervous system and an increased brown fat activity that leads to the reduction of blood cholesterol levels. I would like to thank our colleagues who supported my work continuously, from Page, Professor Miklos Székely, who started my career on this path, and Peter Hegyi, who introduced me to translational research methods, including meta-analysis, Professors Erika Pintér and Zsuzsanna Helyes, who have been continuously supporting my work throughout my career, and from our Department of Thermophysiology, the first author of the paper, Leonardo Kelava, and another co-author, Eszter Pákai, and also Christian Klonfár, who is a student in the program and contributed to previous and ongoing projects. I thank our international collaborator, Andrei Romanovsky, who is also international mentor in the program. And again, I would like to thank the National Academy of Scientist Education for inviting me here today and for selecting our paper for this award. And thank you very much for your attention.